The overall goal of the following experiment is to perform a high-throughput measurement of olfactory receptor activation via a luciferase assay. This is achieved by transfecting Hana-3A cells with vectors containing accessory proteins, firefly and ranilla luciferase, and olfactory receptors in order to express a different olfactory receptor in each well of a 96-well plate. As a second step, the cells are stimulated with odors. Next, luciferase substrate is added, causing the cells that produced luciferase enzyme to luminesce, thus functioning as a reporter for olfactory receptor activation. Ultimately, results show the responses of the population of olfactory receptors to the odors based on the luminescence of each well in the plate.
The main advantages of this technique over existing methods, such as adenoviral transduction of native neurons or calcium imaging, is that this technique both scales to high-throughput screens and utilizes the native cAMP-mediated second messenger pathway to examine olfactory receptor activation.
This method can help answer key questions in the field of olfaction, such as matching odor ligands to olfactory receptors and examining the effect of genetic variation on olfactory receptor activation.
The Hana-3A cells used in this protocol are maintained in M10 media at 37 degrees Celsius with 5% CO2 and should be 100% confluent in order to plate cells for transfection. Aspirate media from a 10 cm dish of cells. Wash cells by adding 10 milliliters of PBS, swirling the dish, and aspirating the PBS. Add 3 milliliters of 0.